Hello there, and welcome back to Bedtime Science. I am really glad you are here tonight. Somewhere in your body, a cell is preparing to divide. It may be a cell near the base of a hair follicle. It may be a cell renewing the lining of your intestine. It may be a stem or progenitor cell in bone marrow preparing to produce another generation of blood cells. Before that cell can become two cells, it has a quiet problem to solve. It must copy almost all of its nuclear DNA, not a few pages of information, not a library shelf. In a typical human cell with two sets of chromosomes, the nuclear DNA contains roughly 6 billion base pairs. If the DNA molecules in that one nucleus were stretched into thin lines instead of folded and packaged, they would extend for roughly 2 meters. Yet the nucleus itself is only a few micrometers across. The copy must be made with extraordinary accuracy. The DNA must remain organized while it is being duplicated. Breaks must be repaired. The two completed sets must then be separated so that each daughter cell receives the chromosomes it needs. And almost none of this requires your awareness. You do not feel a chromosome being copied. You do not notice a repair enzyme recognizing a damaged base. You do not hear a gene being transcribed or a ribosome translating an RNA molecule into protein. The molecular activity inside you is almost perfectly silent from the point of view of consciousness. That makes DNA easy to imagine as a static code stored in a microscopic vault. But DNA is not static. It is copied, bent, twisted, unwound, chemically modified, repaired, rearranged in certain developing immune cells, read into RNA, folded into three-dimensional neighborhoods, packed tightly in some regions, and opened in others. Pieces of it have arrived from ancient viruses. Some sequences have duplicated. Others have disappeared. Some genes have remained recognizable for hundreds of millions of years. Others are recent variations on older themes. And the DNA sequence alone does not tell the whole story of a cell. A neuron and a liver cell contain nearly the same inherited nuclear genome, yet they behave so differently that, seen only through their functions, they might appear to belong to different organisms. One conducts electrical signals through long projections. The other manages metabolism, detoxification, protein production, and the chemistry of blood. Their difference comes not mainly from having different genes, but from using different parts of a largely shared genome in different ways. Tonight, we will follow that quiet system from the molecule outward. We will begin with the strange physical problem of fitting meters of DNA into a microscopic nucleus. We will watch the double helix open and copy itself. We will see how a short stretch of DNA becomes RNA, and how RNA can guide the construction of protein. We will follow chromosomes through meiosis and see why you did not receive a simple half copy of either parent, but a shuffled combination created by recombination and chance. We will meet mutations, most of them ordinary and harmless, that slowly make cells and generations different from one another. We will see how the genome carries traces of ancient migrations and encounters with other human populations. We will see how mobile DNA and ancient viral sequences became permanent residents of our chromosomes. And then we will arrive at the modern age of genetics, when scientists learned not only to read DNA, but in selected situations to alter it. Sequencing, PCR, ancient genomes, cancer genomes, CRISPR, gene therapy. The story will become powerful enough to change medicine, but it will keep returning to one simple fact. Inside a living cell, DNA is never only a code. It is a molecule in motion. The first surprise is how ordinary its ingredients are. DNA stands for deoxyribonucleic acid. A DNA strand is built from repeating units called nucleotides. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases: adenine, thymine, guanine, or cytosine. Their initials give us the familiar alphabet A, T, G, and C. The sugar and phosphate groups form a repeating backbone. The bases point inward, where they can interact with bases on a second strand. Adenine pairs most often with thymine. Guanine pairs with cytosine. The two strands run in opposite chemical directions, making them antiparallel, and together they twist into the shape we call the double helix. It is a beautiful structure, but beauty is not what makes it biologically important. The important feature is complementarity. If one strand contains the sequence A, C, G, T, then the opposite strand is constrained to contain T, G, C, A. Each strand carries enough information to guide construction of its partner. That simple relationship makes accurate copying possible. The double helix was not discovered in one sudden moment by one person. The path took decades. In 1869, a young Swiss scientist named Friedrich Miescher was studying white blood cells collected from used surgical bandages. He isolated an unusual phosphorus-rich substance from cell nuclei. He called it nuclein because of where he found it. Miescher did not know that this material carried hereditary information. At the time, proteins seemed like more plausible candidates. Proteins were chemically diverse. DNA appeared almost monotonous. The mystery deepened in the 20th century. In 1928, Frederick Griffith was studying bacteria that cause pneumonia. He worked with two forms of Streptococcus pneumoniae. One had a smooth protective capsule and could cause disease in mice. Another lacked that capsule and was much less dangerous. Griffith discovered that material from heat-killed virulent bacteria could somehow transform living harmless bacteria, giving them the ability to produce the capsule and become virulent. Something had carried biological information from dead cells into living ones. Griffith did not know what it was. In the 1940s, Oswald Avery, Colin MacLeod, and Macklin McCarty purified the transforming material and showed that destroying proteins did not eliminate the effect, while destroying DNA did. Their conclusion was radical for its time. DNA itself could carry inherited information. Then, in 1952, Alfred Hershey and Martha Chase used viruses that infect bacteria to ask a related question. A bacteriophage is little more than genetic material inside a protein shell. Hershey and Chase labeled viral DNA and viral protein with different radioactive markers. After the viruses infected bacteria, the DNA label entered the cells far more directly than the protein label. Again, DNA behaved like the material of heredity. By then, the question was shifting. If DNA was the information-bearing molecule, what structure could explain its stability and its ability to copy itself? Several clues converged. Erwin Chargaff had measured DNA from different organisms and found a pattern. The amount of adenine was approximately equal to the amount of thymine. The amount of guanine was approximately equal to the amount of cytosine. Species differed in overall base composition, but those pairwise relationships held. At King's College London, Rosalind Franklin and Raymond Gosling used X-ray diffraction to study fibers of DNA. Their images contained geometric information about the molecule. One image, later called Photo 51, showed a strong cross-shaped diffraction pattern characteristic of a helix. Franklin's measurements also helped constrain the width of the molecule and the spacing of repeating features. Maurice Wilkins was working on DNA at the same institution. At Cambridge, James Watson and Francis Crick were trying to build a physical model that could satisfy the available chemical and structural evidence. Data from Franklin's work, including information shown to Watson without Franklin's direct permission, contributed importantly to the final model. In 1953, Watson and Crick proposed the double helical structure of DNA with paired bases inside and sugar phosphate backbones outside. Papers by Franklin and Gosling and by Wilkins and colleagues appeared in the same issue of Nature and supplied experimental evidence central to the structure. The famous model did more than describe shape. It contained a mechanism. If a pairs with T and G pairs with C, then separating the strands creates two templates. Build a complementary strand along each old strand, and one DNA molecule becomes two. Watson and Crick ended their short paper with an understated sentence noting that the specific pairing they proposed immediately suggested a possible copying mechanism. The possibility was elegant. Cells still had to perform it, and performing it across billions of base pairs, every time a dividing cell duplicates its genome, is one of the quietest large engineering tasks in biology. The cell does not begin by unzipping an entire chromosome from one end to the other. Human chromosomes are far too large for that. Replication begins at many sites called origins. From these origins, sections of DNA open and replication forks move outward. Helicase enzymes separate the two strands. Proteins stabilize the exposed single-stranded DNA. Primase makes short RNA primers, because the main replicative DNA polymerases cannot simply begin a new strand from nothing. DNA polymerase then extends from those primers, adding nucleotides according to complementarity. But there is a geometrical complication. DNA polymerases synthesize new DNA only in one chemical direction, conventionally written from 5' to 3'. Because the parental strands run antiparallel, the two new strands cannot be built in exactly the same way at a moving replication fork. One, the leading strand, can be synthesized more or less continuously in the same direction that the fork opens. The other, the lagging strand, is synthesized discontinuously in short pieces called Okazaki fragments. Each fragment begins with a new primer. The RNA primers are later removed. DNA fills the gaps, and DNA ligase seals the remaining breaks in the backbone. The names can sound mechanical because the process is mechanical at a molecular scale. A fork moves, strands separate, enzymes bind and release. DNA loops so that different polymerases can remain coordinated. Tension created by unwinding is managed by topoisomerases that temporarily cut and rejoin DNA to relieve twisting stress. Replication is not perfectly accurate. No copying process is, but cells combine several layers of error control. DNA polymerases select complementary nucleotides with high specificity. Many polymerases proofread the newly made strand and remove mismatched bases. Mismatch repair systems inspect DNA after replication and correct errors that escaped proofreading. Other repair pathways handle damaged bases, chemical modifications, bulky lesions, single-strand breaks, and double-strand breaks. The result is not a mutation-free genome. It is a genome in which copying errors are made rare enough for organisms to survive and reproduce, while variation still accumulates across cells and generations. That balance matters. If mutation rates were dramatically higher, essential information would be lost too quickly. If variation never occurred at all, evolution would lose one of its major sources of novelty. The genome survives because it is stable. Life changes because it is not perfectly stable. Before the DNA can be copied or read, however, it must first exist inside a nucleus without becoming an impossible tangle. Imagine placing several meters of very fine thread inside a small bead, then asking the thread to remain organized enough that any selected region can be found, opened, copied, repaired, and later folded away again. Cells solve this problem with proteins called histones. A segment of DNA wraps around a group of histone proteins to form a structure called a nucleosome. Roughly 147 base pairs curve around each histone core. Under an electron microscope, a lightly packed stretch of chromatin can resemble beads on a string. Although the living structure is more dynamic than that familiar image suggests. Nucleosomes do not merely save space. They help control access. DNA wrapped tightly around histones is harder for many proteins to reach. Chemical modifications to histones can change how strongly nucleosomes interact with DNA or with other proteins. Chromatin remodeling complexes can slide, remove, or reposition nucleosomes. The DNA itself can be methylated at selected sites. Together, these mechanisms help determine whether a regulatory region is accessible or hidden. This is one reason the old metaphor of DNA as a book can mislead. A book lies open or closed as one object. A genome contains thousands of regions that can be opened, closed, looped, contacted, insulated, copied, or silenced independently. A cell does not read its genome from page 1 to page 3 billion. It continuously chooses small regions according to cell type, developmental state, signals, and need. At a larger scale, chromatin folds into loops and domains. Regions that are far apart along the linear DNA sequence can become neighbors in three-dimensional space. An enhancer may influence a promoter tens or hundreds of thousands of base pairs away because folding brings them into physical contact. Other boundaries reduce unwanted interactions between regulatory neighborhoods. Inside the nucleus, therefore, location matters. Genes are not floating independently in a bag of DNA. Their activity depends partly on architecture. During most of a cell's life, chromosomes are not shaped like the compact X forms familiar from school diagrams. Those X-shaped structures are most obvious after DNA replication, when each chromosome consists of two sister chromatids and the chromatin condenses strongly for cell division. Between divisions, chromosomes occupy more extended territories inside the nucleus. Human cells that contain a typical diploid nuclear genome have 46 chromosomes, arranged as 23 pairs. One member of each pair was inherited through an egg and the other through a sperm. 22 pairs are autosomes. The remaining pair contains the sex chromosomes, whose combinations and biological effects are more varied than a simple diagram can capture. Not every human cell follows the typical pattern. Mature red blood cells lose their nuclei as they develop, allowing more internal space for hemoglobin and making them flexible enough to pass through narrow capillaries. Platelets are fragments shed from much larger marrow cells and do not contain a nucleus either. Some cells contain more than one nucleus. Some acquire chromosome number changes. Cancer cells can have profoundly rearranged genomes. So when we say that the human genome is present throughout the body, the accurate picture is not that every one of tens of trillions of cells contains an identical library. Most nucleated cells begin with closely related copies of the inherited genome. From there, development, mutation, rearrangement, and cell specialization create differences. The ends of linear chromosomes create another unusual problem. A circular bacterial chromosome can, in principle, be copied around its entire loop. A linear chromosome has physical ends, and the ordinary replication machinery cannot fully copy the extreme end of one newly synthesized strand after the final RNA primer is removed. Without a solution, chromosomes would become shorter at every division. The solution is a repetitive structure called the telomere. Human telomeres contain repeated DNA sequences bound by specialized proteins. They protect chromosome ends from being mistaken for broken DNA and provide a buffer against the end replication problem. In certain cells, an enzyme called telomerase extends telomeric DNA, using an RNA component inside the enzyme as a template. Telomerase is active in germ cells, some stem and progenitor cells, and many cancers. In most ordinary somatic cells, telomerase activity is limited, and telomeres tend to shorten across repeated divisions. When they become critically short or dysfunctional, cells can enter senescence or die. This has made telomeres famous in discussions of aging. But they are not a single countdown clock that determines the age of a person. Aging involves DNA damage, epigenetic change, mitochondrial function, protein quality control, immune regulation, cellular senescence, stem cell behavior, metabolism, tissue structure, and many other interacting processes. Telomeres are one part of the biology, not a simple meter of remaining life. Inside a stable chromosome, most DNA is not being copied at any given moment. Much of it is instead waiting to be used. To use genetic information, a cell often begins by making RNA. RNA resembles DNA in some ways. It is also a nucleic acid built from nucleotide subunits. But RNA usually contains the sugar ribose rather than deoxyribose, and it uses uracil in place of thymine. RNA molecules are often single-stranded and can fold into intricate shapes. Some carry messages. Some form structural parts of molecular machines. Some catalyze reactions. Some regulate other RNAs or genes. The process of copying information from DNA into RNA is called transcription. A protein-coding gene is not simply a stretch of DNA that a cell reads whenever it wishes. It sits within a regulatory landscape. Nearby promoter sequences help position the transcription machinery. Enhancers can raise activity from a distance. Repressors can reduce it. Transcription factors bind selected DNA sequences and respond to hormones, nutrients, stress signals, developmental programs, and signals arriving from other cells. When the appropriate combination is present, RNA polymerase begins moving along one DNA strand. The DNA opens locally. The polymerase reads the template strand and assembles a complementary RNA molecule. Where the DNA has an A, the RNA may receive U. Where the DNA has a C, the RNA receives G. The growing RNA peels away and the DNA strands close again behind the enzyme. For many human protein-coding genes, the first RNA product is not yet ready to be translated. It must be processed. A modified structure called a 5' cap is added near one end. A poly-A tail is added at the other. Introns are removed and exons are joined by a large RNA protein machine called the spliceosome. The words intron and exon can sound as though one part is meaningless and the other meaningful. Reality is more interesting. Many introns contain regulatory information or functional elements. Exons can include untranslated regions as well as protein coding sequence. And the cell does not always join the same exons in the same way. Through alternative splicing, one gene can produce multiple mature RNA forms. Different tissues can favor different combinations. A gene expressed in brain may be spliced differently from the same gene in muscle. The final proteins can therefore differ even though they originated from the same genomic region. This is one reason counting genes does not tell you how many different proteins a human body can produce. Humans have roughly 20,000 protein-coding genes, not hundreds of thousands. Yet alternative splicing, alternative transcription start sites, RNA editing, chemical modification, protein cleavage, and post-translational modification multiply the number of functional molecular forms enormously. The mature messenger RNA can then leave the nucleus through a nuclear pore. Outside, it meets a ribosome. Ribosomes are among the oldest and most important molecular machines in life. They are made from proteins and ribosomal RNA, and the catalytic heart that creates peptide bonds is built from RNA. This is one clue that RNA may have played an even more central role early in the history of life, before the modern division of labor among DNA, RNA, and protein became established. A ribosome reads messenger RNA 3 bases at a time. Each group of 3 is a codon. With 4 possible RNA bases and 3 positions, there are 64 possible codons. Most specify one of the 20 standard amino acids. 3 are commonly used as stop signals. AUG usually encodes methionine and often marks the beginning of translation, although the rules of initiation involve more than recognizing one triplet. Transfer RNAs solve the translation problem by connecting two molecular languages. One part of a transfer RNA contains an anticodon that can pair with a codon and messenger RNA. Another part carries a particular amino acid. Enzymes called aminoacyl tRNA synthetases attach the correct amino acid to the correct transfer RNA. The ribosome then moves along the messenger RNA, selecting transfer RNAs according to codon pairing and joining their amino acids into a growing chain. The sequence of bases has become a sequence of amino acids. But even then, information has not become function automatically. A newly synthesized protein must fold. Some fold largely by their own chemical properties. Others are helped by chaperone proteins. Some receive sugars, phosphate groups, lipids, or other modifications. Some are cut into active forms. Some are transported into the nucleus, mitochondria, cell membrane, lysosome, or outside the cell. A protein that folds incorrectly may be refolded or destroyed. Cellular life is therefore not a simple line from gene to trait. DNA influences RNA. RNA influences protein. Proteins regulate DNA and RNA. Metabolites alter enzymes. Hormones alter transcription. Cell shape influences signaling. Mechanical forces influence gene expression. The environment around a cell can change which parts of the genome are used. Francis Crick's central dogma is often summarized as DNA makes RNA makes protein. That summary is useful if it is not taken too literally. The central dogma was fundamentally about restrictions on sequence information transfer, especially the idea that sequence information does not flow from protein back into nucleic acid. Biology contains reverse transcription from RNA into DNA. It contains RNA genomes. It contains functional RNAs that are never translated. It contains proteins that alter chromatin and thereby change future gene expression. The cell is a network, not a one-way conveyor belt. The genetic code itself carries another kind of history. With a few important exceptions, organisms across the tree of life use almost the same mapping between codons and amino acids. Bacteria, oak trees, mushrooms, whales, and humans all interpret most of the same three-letter RNA words in the same way. Mitochondria and some microorganisms use modified versions, but the broad conservation is remarkable. This near universality suggests that the basic genetic code became established very early, before the major lineages of modern life diverged. It also makes genetic engineering possible in a practical sense. A human protein coding sequence can be placed into bacteria or yeast, and with appropriate regulatory adjustments, those cells can often produce the human protein because their ribosomes interpret the codons through nearly the same ancient dictionary. Recombinant human insulin is one famous result of that shared molecular language. The code is redundant, meaning several codons can specify the same amino acid, but redundancy does not make synonymous changes automatically meaningless. Different organisms and tissues can prefer different codons. A nucleotide change can alter RNA structure, splicing, stability, or the speed of translation even when the amino acid sequence remains unchanged. Once again, the simplest classroom rule survives, but the living system adds another layer around it. The central role of RNA has also inspired an idea about life's earliest history called the RNA world hypothesis. Modern cells divide labor among DNA, RNA, and proteins. DNA is especially useful for stable information storage. Proteins perform an enormous range of chemistry. RNA often carries information between them, but RNA can do both kinds of work. It can store sequence information, and some RNA molecules can catalyze chemical reactions. The catalytic core of the ribosome is itself ribosomal RNA. Self-splicing RNAs and other ribozymes demonstrate that RNA can act as an enzyme. These observations support the possibility that, before modern DNA genomes and protein enzymes evolved, earlier biological systems relied more heavily on RNA-like molecules for both inheritance and catalysis. No intact RNA world survives for us to inspect, and researchers still debate how the first self-replicating chemical systems emerged, how nucleotides formed under early Earth conditions, and how RNA, peptides, membranes, and metabolism became linked. But the hypothesis gives the ribosome a different atmosphere. Every time one of your cells translates a messenger RNA, it may be using a molecular architecture that preserves echoes of a much older stage in the history of life. And now we can ask a deeper question. If almost every nucleated cell in one body contains nearly the same inherited DNA sequence, how did that one sequence produce a retina, a heart, a liver, skin, bone, and a brain? The answer begins before birth, when one cell becomes two, then four, then many, and cells gradually acquire different histories even while sharing almost the same genome. A human life begins, biologically, with a remarkable compression of possibility. One fertilized egg contains chromosomes from two parents and the molecular machinery needed to begin development. It does not contain a tiny preformed body. It contains a genome, inherited molecules already present in the egg, and a system capable of changing itself step by step. The first divisions increase cell number without immediately creating the full diversity of adult tissues. Then small differences begin to matter. A cell may receive a stronger signal from one neighbor than another. It may occupy a slightly different position. A transcription factor may become active. That factor may activate another gene while repressing a third. Chromatin may open around one developmental program and close around another. Gradually, temporary differences become stable cell identities. This process is called differentiation. The remarkable part is that differentiation usually happens without deleting most of the genes a cell no longer needs. A neuron does not become a neuron by throwing away liver genes. A liver cell does not become a liver cell by removing genes used in the retina. Instead, cells establish regulatory states that make some genetic programs accessible and others difficult to activate. This is where epigenetics enters the story. The word is used in several ways, which is one reason it can cause confusion. In a careful biological sense, epigenetic regulation refers to stable or sometimes heritable changes in gene activity and chromatin state that do not require changes in the underlying DNA sequence. DNA methylation is one important mechanism. Histone modifications are another. Chromatin remodeling, nucleosome placement, and regulatory RNAs can also contribute. DNA methylation in mammals commonly involves adding a methyl group to cytosine bases, especially at CPG sites. Heavy methylation near certain promoters is often associated with reduced transcription, although the relationship depends on genomic context. Histone acetylation is often associated with more accessible chromatin, while different forms of histone methylation can mark active or inactive regions depending on which amino acid is modified. The popular version of epigenetics sometimes goes much further. It may suggest that every experience writes a lasting molecular memory onto DNA, that these marks freely pass to children, or that behavior can reliably rewrite inherited biology across generations. That is too simple. Cells certainly change gene expression in response to environment. Nutrition, hormones, stress, inflammation, toxins, learning, aging, and many other influences can alter transcription and chromatin. Some epigenetic states persist through cell division. In plants and some animals, environmentally influenced epigenetic states can sometimes persist across generations. In mammals, however, much of the epigenome is extensively reprogrammed during formation of eggs and sperm and again after fertilization. Some marks escape this resetting, and there are well-established forms of epigenetic inheritance such as genomic imprinting. But broad claims that ordinary life experiences are routinely transmitted for many human generations through stable epigenetic marks remain much less secure. The cautious version is already extraordinary enough. Your cells remember what kind of cells they are partly through molecular states layered on top of DNA sequence. One of the clearest examples is genomic imprinting. For most autosomal genes, the maternal and paternal copies are potentially active. In a small subset, however, expression depends on which parent supplied the chromosome. One parental copy is active while the other is silenced or greatly reduced through epigenetic marks established in the germline. This creates an unusual situation. Two DNA sequences can be nearly identical, yet their parental origin changes whether they are used. The chromosome 15 region involved in Prader-Willi syndrome and Angelman syndrome demonstrates the principle. Disruptions in overlapping genomic territory can produce very different disorders depending on whether the affected material came from the father or the mother, because different imprinted genes are active from each parental copy. Imprinting also shows why the genome cannot be understood as sequence alone. History matters. The same idea appears on a much larger scale with the X chromosome. In many cells of people with two X chromosomes, one X becomes largely inactive early in development. This dosage compensation process prevents many X-linked genes from being expressed at roughly twice the level found in cells with one X. The choice is largely random in embryonic cells. If one early cell inactivates the X inherited from one parent, many of its descendants tend to maintain that choice. A neighboring early cell may inactivate the other X. The adult body therefore becomes a mosaic of cell lineages with different active X chromosomes. The inactivated X condenses into a structure historically called a bar body, and a long non-coding RNA called XIST spreads across the chromosome and helps establish silencing. The silencing is not absolute. Some genes escape X inactivation. Patterns can vary by tissue and individual, but the broad process reveals something profound about development. A cell can silence almost an entire chromosome and maintain that state through thousands of later divisions without changing the DNA sequence itself. Development uses genetic sequence as a set of possibilities and regulatory systems as a way to choose among them. Some developmental genes are especially powerful because they control large networks below them. The HOX genes are a famous example. They encode transcription factors that help establish positional identity along the head-to-tail axis of an embryo. Related HOX genes pattern bodies across much of the animal kingdom. A gene involved in positioning structures in a fruit fly has recognizable relatives in mammals. This deep conservation is one of the strongest reminders that your genome was not invented for humans. It is a modified inheritance from earlier life. The genes that help cells copy DNA, make ribosomes, repair damage, control the cell cycle, transport molecules, and communicate with neighbors often have ancient relatives in organisms separated from us by vast evolutionary distances. Evolution usually works by modifying existing systems. A gene can duplicate. One copy continues its old role while the other accumulates changes. A regulatory sequence can shift the time or place a gene is expressed. A protein domain can be combined with another. A mobile genetic element can bring new regulatory sequences. An old pathway can be recruited for a new developmental function. The history of life becomes layered rather than replaced. Even development itself depends on selective destruction as well as growth. During formation of hands and feet, cells between developing digits die through programmed cell death. In the nervous system, more connections may initially form than will remain, and activity helps refine them. Immune cell development removes or restrains many cells with dangerous receptor specificities. Genes control not only what is built, but what is removed. The genome does not contain one instruction that says build a body. It contains interacting molecular systems that create local decisions. Turn this gene on here. Keep that region silent there. Divide. Stop dividing. Move. Attach. Differentiate. Die. Respond to this signal only if another signal is also present. From those local rules, anatomy emerges. And once an organism is formed, its cells must preserve identity while remaining flexible enough to respond. A liver cell exposed to a hormone may change the expression of hundreds of genes within minutes or hours. An immune cell encountering a pathogen can reorganize its transcriptome dramatically. A muscle cell responds to repeated exercise by changing metabolism and gene regulation. A neuron changes the strength of synapses after experience while remaining a neuron. Stability and flexibility exist together. The same is true across generations. Parents transmit chromosomes, but those chromosomes are not passed forward as untouched blocks. Before an egg or sperm is formed, a special kind of cell division shuffles them. That process is meiosis. And meiosis is one reason siblings from the same parents can be so different. Ordinary cell division, mitosis, is mainly a copying process. A cell duplicates its chromosomes and distributes sister chromatids so that the two daughter cells receive closely matched sets. Meiosis has a different purpose. It produces cells with one set of chromosomes rather than two, preparing for fertilization, when two haploid sets will come together again. Before meiosis begins, the DNA is replicated. Each chromosome now consists of two sister chromatids. But instead of immediately separating those sisters, homologous chromosomes pair with one another. The chromosome inherited from one parent aligns with its corresponding chromosome inherited from the other. They are similar, but not identical. At many positions, they carry the same DNA sequence. At others, they carry different variants. During prophase 1 of meiosis, homologous chromosomes can exchange corresponding segments through crossing over. DNA molecules break and rejoin so that a chromatid can emerge with one part derived from one grandparent and another part derived from the other. The exchange is not random at the scale of individual bases. Recombination is shaped by chromosome structure, sequence, sex, and specialized proteins. Some genomic regions are recombination hotspots. Others recombine rarely. But from the perspective of ancestry, the effect is a continual shuffling. You did not inherit one intact chromosome, one from a maternal grandmother, and one intact chromosome, one from a paternal grandfather. The chromosomes passed through your parents had already been mosaics created by earlier generations of recombination. Your own eggs or sperm, if you produce them, would contain another round of newly shuffled mosaics. Then homologous chromosome pairs orient at the first meiotic division. For each pair, which homologue goes toward which future cell is largely independent of how other pairs orient. This independent assortment creates enormous combinatorial variety even before recombination is considered. After two divisions, one starting cell can produce haploid products carrying different combinations of chromosomes and recombinant chromatids. Fertilization adds another layer of chance. One egg meets one sperm out of an enormous number of possibilities. The resulting genome is therefore not a simple average of two parental genomes. It is one realization from a vast field of possible combinations. Gregor Mendel discovered the statistical shadow of this process before anyone knew what chromosomes or DNA were. In the 1850s and 1860s, working with pea plants, Mendel performed controlled crosses involving traits that could be distinguished clearly: seed shape, seed color, flower color, plant height, pod form. His most important achievement was not simply breeding peas. It was counting. Mendel tracked traits across generations and recognized numerical patterns. A trait that disappeared in one generation could reappear in the next. The ratios suggested that hereditary information came in discrete units rather than blending permanently like two colors of paint. For one trait, an organism could carry two hereditary factors, one from each parent. Those factors separated during formation of reproductive cells. Fertilization restored the pair. We now call the factors alleles of genes, and we understand their separation through chromosome behavior during meiosis. Mendel's law of segregation remains one of the foundations of genetics. His law of independent assortment also captures a real principle, but with an important condition. Genes on different chromosomes can assort largely independently. Genes far apart on the same chromosome can also appear nearly independent because crossing over frequently separates them. But genes close together on one chromosome tend to travel together. They are linked. This discovery helped connect genes to chromosomes. In the early 20th century, Thomas Hunt Morgan and colleagues used fruit flies to show that some traits were inherited together more often than Mendel's simple, independent model predicted. The frequency with which linkage was broken by recombination could be used to estimate relative distances between genes. The first genetic maps were therefore not maps of DNA letters. They were maps of probability. If two genes were frequently separated by recombination, they were probably farther apart. If they were rarely separated, they were probably closer. Modern sequencing can read the DNA sequence directly, but the old maps captured the chromosome's behavior before the molecule could be seen in detail. One familiar human example of alleles is the ABO blood group. The ABO gene encodes an enzyme that modifies carbohydrate structures on the surface of red blood cells and other tissues. Common A and B alleles produce enzymes with different substrate specificity. A common O allele produces an inactive form, so the precursor carbohydrate remains unmodified in the same way. A and B are codominant. A person inheriting one A allele and one B allele can express both A and B antigens. O is usually recessive to functional A or B alleles. The familiar four blood groups emerge from these allele combinations. But even this apparently simple system is surrounded by complexity. Blood transfusion compatibility also depends on the Rh system and many other blood group antigens. Rare ABO variants exist. The antibodies involved develop through interactions between the immune system and environmental antigens. Medical genetics quickly becomes more detailed than a classroom Punnett square. The Punnett square remains useful because it teaches probability. Suppose two parents each carry one typical copy and one disease-causing recessive copy of a gene. For each pregnancy, the probabilities may be 1/4 for inheriting two typical copies, 1/2 for inheriting one of each, and 1/4 for inheriting two disease-associated copies. But those probabilities do not remember previous children. If the first child inherited two recessive alleles, the next pregnancy does not become safer because the family has already used its 1/4 outcome. Each conception is a new sampling event. And most human traits do not follow such clean single gene rules at all. Height is not controlled by one tall gene and one short gene. Body mass is not controlled by one weight gene. Coronary artery disease, type 2 diabetes, asthma, schizophrenia, and many other common conditions involve large numbers of genetic variants interacting with development, environment, behavior, age, and chance. This is polygenic inheritance. A genome-wide association study, or GWAS, searches large groups of people for statistical associations between genetic variants and traits. If one variant is slightly more common among people with a condition than among comparison participants, the region may contain or tag a biological influence on risk. Modern studies can include hundreds of thousands or millions of participants. They have identified thousands of associated loci for complex traits. The effect of a single common variant is often small. That can feel disappointing if one expects genetics to reveal one decisive cause. But biology rarely owes a complex human trait to one switch. Many pathways contribute. A variant may alter gene expression by a few %. Another changes a receptor slightly. Another affects development in one tissue. Another matters only under a particular diet, exposure, infection history, or hormonal state. The effects accumulate and interact. A polygenic score attempts to combine information from many variants into a statistical estimate of genetic predisposition. Such scores can be useful in research and, for selected conditions, may contribute to risk prediction. But they are not prophecies. Their accuracy depends strongly on the populations used to build them, the trait being studied, and the clinical context. A score cannot capture every environmental influence, rare variant, structural variant, developmental event, or future exposure. This leads to one of the most misunderstood words in genetics: heritability. Heritability does not mean the percentage of one person's trait caused by genes. It is a population statistic. In a particular population living in particular environments, heritability estimates how much of the observed variation in a trait is statistically associated with genetic variation. A highly heritable trait can still be strongly influenced by environment. Imagine a population in which everyone receives nearly identical nutrition. Genetic differences may explain much of the remaining variation in height. Change the nutritional environment dramatically, and average height can change even though the genome of the population barely changes. Conversely, a trait can have low heritability in one setting because environmental differences dominate, even if genes are biologically necessary for the trait to exist. Genes and environment are not rival explanations dividing a body between them. Genes build systems that respond to environments. Environments influence gene expression and development. The effects are intertwined from the first cell onward. Even a classical single-gene condition can vary between people carrying the same major mutation. Other genes can modify severity. Environmental factors can alter symptoms. Random events during development can matter. Different tissues may carry different proportions of mutant and normal cells. One striking example is phenylketonuria, or PKU. Variants affecting the enzyme phenylalanine hydroxylase can cause phenylalanine to accumulate to harmful levels, especially in the developing nervous system. The genetic cause can be inherited, yet the outcome can be transformed by environment. Newborn screening and a carefully controlled diet can prevent much of the severe neurological damage that would otherwise occur. A genetic condition is not therefore proof that biology is destiny. Sometimes knowing the genetic mechanism reveals exactly where intervention becomes possible. The same principle appears in sickle cell disease. A single nucleotide change in the beta-globin gene alters one amino acid in beta-globin. That molecular substitution changes how hemoglobin molecules behave when oxygen levels fall. Hemoglobin can polymerize, red blood cells can become rigid and sickle-shaped, blood flow can be obstructed, and repeated episodes can damage organs. One DNA letter can matter enormously. Yet even sickle cell disease is not one uniform outcome. Fetal hemoglobin levels, other genetic modifiers, infection, oxygenation, treatment, access to care, and many other factors influence severity. The mutation is central. The phenotype is the mutation living inside an entire organism and environment. This is the pattern genetics keeps teaching. A genome influences possibility. A life is what happens when those possibilities meet development, history, and the world. and the genome itself does not remain perfectly unchanged during that life. Every time DNA is copied, every time it is exposed to reactive chemistry, every time sunlight strikes skin or a cell repairs a broken chromosome, there is a possibility of alteration. Those alterations are mutations. The word mutation often sounds dramatic because mutations are frequently introduced through disease. In biology, the word is neutral. A mutation is a change in DNA sequence. It can be harmful, helpful, or effectively irrelevant. Most changes in a genome do not create a visible new trait. Some occur in regions where they have little measurable effect. Some are corrected before they become permanent. Some alter gene regulation subtly. A small fraction can have large consequences. Mutations also come in many sizes. A single nucleotide can be replaced by another. One or a few bases can be inserted or deleted. A segment can be duplicated. A chromosome can invert a piece of itself. Material can move from one chromosome to another. Copy number can increase or decrease. Whole chromosomes can be gained or lost. When a mutation happens matters as much as what the mutation is. A change in a sperm or egg lineage can be transmitted to a child. A change arising in a skin cell usually remains within the descendant lineage of that skin cell. If a mutation occurs very early after fertilization, a large fraction of the body may carry it. If it occurs later, it may be confined to one tissue or even one small clone of cells. This creates somatic mosaicism. The cells in one person are not all genetically identical. You began from one fertilized egg, but every later cell division created an opportunity for new changes. Over a lifetime, different lineages accumulate different mutations. Most never become noticeable. Some contribute to aging or disease. Some can be detected only with sensitive sequencing. Even monozygotic twins begin with the same fertilized egg and then follow separate developmental histories. Post-zygotic mutations can arise after the embryo divides, leaving one twin with variants not present in the other, or leaving variants in different proportions of their tissues. They are extraordinarily similar genetically. They are not frozen copies that remain molecularly identical forever. The sources of mutation are everywhere because chemistry itself is imperfect. DNA bases can change spontaneously. Reactive oxygen species generated during normal metabolism can damage nucleotides. Ultraviolet light can create covalent links between neighboring bases. Ionizing radiation can break DNA strands. Tobacco smoke contains chemicals that form DNA adducts. Some viruses alter cell growth or integrate genetic material. Replication occasionally inserts the wrong nucleotide. Cells spend enormous effort repairing this damage. Base excision repair removes small chemically altered bases. Nucleotide excision repair removes larger distortions in the helix. Mismatch repair corrects errors left after replication. Double-strand breaks can be repaired by pathways that directly join DNA ends or use a homologous template. Each pathway solves a different problem and makes different compromises. Repair itself can occasionally introduce change. Non-homologous end joining can reconnect a dangerous double-strand break quickly, but may add or remove a few bases at the junction. Homologous recombination can be more precise when a suitable template is available, usually a sister chromatid in certain parts of the cell cycle. Inherited defects in repair pathways reveal how important this constant maintenance is. People with xeroderma pigmentosum have defects in nucleotide excision repair and can be extraordinarily sensitive to ultraviolet light, with greatly increased risk of skin cancer. Lynch syndrome can arise from inherited defects in mismatch repair genes, increasing the accumulation of mutations in certain tissues and elevating cancer risk. Variants in BRCA1 or BRCA2 can impair homologous recombination repair and increase risk for several cancers. Cancer is, at its core, an evolutionary process occurring within the body. A cell acquires changes that allow it to divide more, ignore growth restraints, resist death, alter metabolism, evade immune control, invade tissue, or survive in new environments. The expanding clone then creates more cells in which additional variation can arise. Different subclones compete under pressures created by oxygen, nutrients, immunity, and treatment. No single mutation is called cancer in isolation. Cancer usually requires a series of changes affecting multiple systems. Some alterations activate proto-oncogenes, turning normal growth-promoting genes into excessively active oncogenes. Others disable tumor suppressor genes that ordinarily restrain division, detect damage, or trigger cell death. Still others alter DNA repair, chromatin regulation, metabolism, or communication with surrounding tissue. The genome of a tumor can therefore look very different from the inherited genome measured in blood or saliva. This distinction between germline and somatic genetics matters in medicine. A person may inherit a variant that increases susceptibility to cancer in every nucleated cell, but the tumor itself acquires many additional somatic mutations. Conversely, a tumor can carry an actionable mutation that is not inherited and does not imply that relatives carry the same risk. Sequencing a cancer can reveal both histories if the tumor is compared with normal tissue. Some cancers even preserve molecular fingerprints of the mutational process that shaped them. Ultraviolet light tends to produce characteristic patterns of base changes. Tobacco-associated carcinogens produce another pattern. Defective mismatch repair creates characteristic mutation profiles. Enzymes that normally edit nucleic acids can leave their own signatures when dysregulated. Researchers call these mutational signatures. They are statistical patterns, not labels stamped on individual DNA molecules, but they can help reconstruct the forces that acted on a tumor over time. The genome becomes a record of damage as well as inheritance. Aging tissues also accumulate clones without necessarily becoming malignant. In blood, for example, a stem cell can acquire a mutation that gives it a modest growth advantage. Its descendants gradually occupy a larger fraction of blood production. This age-associated clonal hematopoiesis can occur in people who have no leukemia. It may increase future risk of blood cancer or cardiovascular disease, but many people carrying such clones never develop those outcomes. Again, genetics describes probability rather than certainty. Mutation also creates the raw material of evolution between generations. Each child carries new germline variants that were not present in either parent's inherited sequence. The number is usually measured in dozens, and it tends to increase with paternal age because sperm-producing cell lineages undergo repeated divisions across adulthood. Most new variants are neutral or nearly neutral. A harmful variant may reduce reproductive success and be removed by selection over time. A beneficial variant can become more common if it improves survival or reproduction in a particular environment. Many variants drift in frequency largely by chance, especially in small populations. Natural selection does not inspect a mutation and decide whether it is useful. Variation appears without foresight. The environment determines which effects matter. A classic human example is the sickle cell allele. Carrying two disease-associated copies can cause severe sickle cell disease. Carrying one copy, however, can reduce the risk of severe malaria in regions where malaria is common. That selective advantage helped maintain the allele at relatively high frequencies in several populations with historical malaria exposure. The same DNA variant can therefore be harmful in one genetic state, beneficial in another context, and nearly irrelevant in a third environment. Lactase persistence offers another example. Most mammals reduce production of the lactase enzyme after weaning. In several human populations with long histories of dairying, regulatory variants evolved independently that maintain lactase expression into adulthood. The variants differ between populations, yet they produce a similar phenotype. Evolution found more than one genetic route to the same dietary adaptation. High-altitude populations provide another lesson. People whose ancestors lived for many generations on the Tibetan Plateau, in the Andes, or in Ethiopian highlands show physiological adaptations to low oxygen, but the genetic solutions are not identical. In Tibetans, variation near genes including EPAS1 contributes to an oxygen response pattern distinct from some Andean adaptations. Part of the Tibetan-associated EPAS1 haplotype appears to have entered modern human ancestors through ancient interbreeding with Denisovan-related people. That means a sequence inherited from another human population tens of thousands of years ago still influences physiology in living people today. The genome is not only a family record, it is an archaeological site. Every chromosome contains layers left by mutation, selection, migration, recombination, population bottlenecks, and encounters between populations that once seemed separate. To read those layers, genetics had to learn how to compare genomes across people, across continents, and eventually across species and extinct humans. Long before whole genomes could be sequenced, researchers used smaller genetic markers to reconstruct population history. Mitochondrial DNA became especially useful. Mitochondria are the energy-converting organelles found in nearly all human cells, and they contain their own small circular genome. The human mitochondrial genome is only about 16,500 base pairs long, tiny compared with the billions of base pairs in the nucleus. It contains 37 genes involved largely in mitochondrial protein synthesis and oxidative phosphorylation. Mitochondrial DNA is inherited overwhelmingly through the maternal line in humans. That inheritance pattern gives it a simple genealogical structure. Your mitochondrial DNA usually came from your mother, whose mitochondrial DNA came from her mother, and so on. Mutations accumulate along those lines, allowing researchers to compare living populations and estimate branching relationships. This work helped support the broader conclusion, also supported by nuclear DNA, fossils, archaeology, and other evidence, that Homo sapiens originated in Africa and later expanded into the rest of the world. The phrase mitochondrial Eve is sometimes misunderstood. It does not describe the first woman, the only woman alive at her time, or the mother of all other people in every genealogical sense. It refers to the most recent woman from whom all living people's mitochondrial lineages descend through an unbroken chain of mothers. Many other women lived at the same time and contributed enormous portions of the nuclear genomes of later humans. Their direct maternal lines simply ended at some point because a branch had no daughters whose maternal line continued to the present. The Y chromosome can be used in a related way for paternal line ancestry in people who inherit a Y chromosome. Again, the most recent common ancestor of living Y lineages was not the only man alive in his time and need not have lived at exactly the same time as the mitochondrial common ancestor. Genetic genealogy is full of these distinctions. A genealogical ancestor and a genetic ancestor are not the same thing. Go back enough generations, and the number of positions in a family tree doubles so rapidly that it exceeds the number of people who actually lived. The tree folds back on itself because the same ancestors occupy many positions. Yet recombination means that some genealogical ancestors eventually contribute no identifiable DNA to a particular descendant. You can descend from someone without carrying a recognizable segment of that person's genome. The farther back we go, the less ordinary pedigree language can tell us about which molecules survived. Ancient DNA transformed this problem by allowing researchers to compare living genomes with DNA recovered from bones and teeth. Ancient DNA is difficult material. After death, DNA breaks into short fragments. Chemical damage accumulates. Microbial DNA invades remains. Modern human DNA can contaminate samples through excavation and handling. Warm, humid environments accelerate degradation, while cold, dry environments can preserve fragments far longer. Early studies could examine only small regions. Improvements in clean laboratories, sequencing, library preparation, and statistical methods eventually made entire ancient genomes possible. One of the most dramatic results concerned Neanderthals. For much of the 20th century, discussions often treated Neanderthals as a side branch that disappeared without contributing to living humans. Ancient genome sequencing changed that picture. Many people whose ancestry lies largely outside sub-Saharan Africa carry a small percentage of DNA inherited from Neanderthal populations. The exact amount varies among people and populations, and some African populations also carry Neanderthal-derived segments through later gene flow from Eurasian populations. The conclusion is simple and profound. When ancestors of modern humans expanded into Eurasia, they did not only replace every other human population they encountered, they sometimes had children with them. Then a small bone fragment from Denisova Cave in Siberia revealed another population. DNA recovered from a finger bone showed that it did not belong to a Neanderthal or a typical modern human. The genome represented a distinct archaic lineage now called Denisovan. Later genetic evidence showed that Denisovan-related ancestry survives at especially high levels in some populations in Oceania and parts of Asia. The discovery was extraordinary because genetics identified an ancient human population before paleontology had anything like a complete skeleton to represent it. A genome had become a fossil. These archaic contributions were not preserved uniformly. Many introgressed sequences were lost. Some were probably harmful in the genetic background of modern humans. Others drifted away by chance. A few contributed useful variants. The EPAS1 region associated with high-altitude adaptation in Tibetans is one of the best-known examples of archaic introgression followed by selection. Other Neanderthal-derived variants have been associated with immune function, skin biology, metabolism, and disease risk, although any one association must be interpreted carefully. A sequence that was useful in an ancient infection environment can have different effects in a modern lifestyle. Population genetics therefore resists clean stories about purity or isolated lineages. Human populations have repeatedly split, moved, mixed, contracted, expanded, and met again. Genetic ancestry is statistical and layered. It does not divide humanity into a small set of sharply bounded biological races. Human genetic variation is often gradual across geography, and much variation is shared across populations. Social identities, ethnicities, languages, and national categories have histories that do not map neatly onto genetic clusters. The genome can reveal migration and relatedness. It cannot provide a simple molecular definition for every human identity. The deeper we look into chromosomes, the more mixed the history becomes. Some of the most ancient residents of the human genome are not even recognizably human in origin. Nearly half of the human genome can be traced to transposable element families and their remnants. Transposable elements are sequences capable, or once capable, of moving or copying themselves to new genomic locations. Some move through AD and A intermediate. Others, called retrotransposons, are copied into RNA and then reverse transcribed back into DNA at a new site. Most copies in the human genome are no longer mobile. They accumulated mutations over millions of years and became molecular fossils. LINE1 elements are among the most abundant. A small number of LINE1 copies remain capable of retrotransposition. Their molecular machinery can also help mobilize other sequences. New insertions occasionally occur in the germline and can sometimes disrupt genes or regulatory regions. But mobile elements are not simply parasites that the genome failed to remove. Evolution can reuse their sequences. A transposable element may bring a transcription factor binding site into a new location. Its promoter can be recruited into a regulatory network. Its sequence can become part of a non-coding RNA. Over evolutionary time, pieces that once served the mobile element can become useful to the host organism. Endogenous retroviruses provide one of the strangest examples. A retrovirus normally inserts a DNA copy of its genome into an infected cell. If such an insertion occurs in a cell lineage that contributes to eggs or sperm and is then inherited, viral DNA can become a permanent part of the host species' genome. Across millions of years, repeated events left human endogenous retroviral sequences scattered through our chromosomes. Together, sequences of endogenous retroviral origin account for roughly 8% of the human genome, although most are fragmented and inactive. Some viral genes were domesticated. Syncytin proteins are derived from ancient retroviral envelope genes. Viral envelope proteins evolved to fuse viral and host membranes. In mammals, related captured genes became important in placental cell fusion and development. A molecular tool once used by a virus to enter cells became part of mammalian reproduction. The distinction between self and foreign becomes less obvious across evolutionary time. Your genome contains genes inherited from ancient vertebrates, sequences shared with single-celled ancestors, mobile elements that copied themselves, and fragments of viruses that entered germlines long before Homo sapiens existed. The genome is not a manuscript written once. It is a manuscript copied by generations of editors who never met, filled with reused paragraphs, duplicated passages, deleted chapters, and annotations whose original purpose has sometimes disappeared. And yet cells read it with remarkable precision. To understand exactly where all those passages lie, scientists needed a technology that genetics lacked for most of its history. They needed to read DNA letter by letter. The first practical methods for reading long stretches of DNA arrived only in the late 20th century. Frederick Sanger and colleagues developed a sequencing method based on controlled termination of DNA synthesis. A DNA polymerase copied a template while occasionally incorporating modified nucleotides that stopped further extension. By separating the resulting fragments according to length, researchers could infer the sequence one base at a time. The original versions used radioactive labels and gels read manually. Automation replaced much of that work with fluorescent dyes, capillary electrophoresis, lasers, and computers. The underlying logic remained recognizable. Make collections of DNA fragments ending at known bases, separate them by length, and reconstruct the sequence. Sanger sequencing made possible one of the most ambitious scientific projects ever attempted. The Human Genome Project formally began in 1990. Its goal was not to sequence one person's genome as a consumer service. It was to build a high-quality reference sequence and develop the technologies, maps, databases, ethical frameworks, and analytical methods needed to make human genomics possible. The task was enormous by the standards of the time. The human genome contains roughly 3 billion base pairs in one haploid set. Sequencing instruments read relatively short pieces. Researchers therefore had to break genomic DNA into many fragments, determine the sequence of those fragments, and assemble them computationally using overlaps and physical maps. It was like reconstructing a library after shredding every book, except many pages contained nearly identical repeated paragraphs. In 2000, researchers announced a working draft. In 2001, major analyses were published. In 2003, The international project declared the reference essentially complete for the regions that available technology could reliably resolve. The achievement was transformative. But the word complete carried a technical meaning. Highly repetitive regions, especially around centromeres, segmental duplications, and other difficult structures, still contained gaps. The 2003 reference covered the great majority of gene-containing sequence with extremely high accuracy, but it was not literally a gapless sequence from one chromosome into the other. That final challenge took nearly 2 more decades. Long-read sequencing technologies changed what could be assembled. Instead of reading only short fragments, newer systems could follow individual DNA molecules across thousands or tens of thousands of bases, sometimes much farther. Long reads could span repeats that had previously been impossible to place. In 2022, the Telomere-to-Telomere Consortium published the first gapless sequence of a human genome assembly, known as T2TCHM13. Even that milestone did not mean that science had discovered the one definitive human genome. There is no single sequence that can represent all humans. Every reference is a coordinate system. The earlier reference sequence drew most of its material from a small number of donors and did not capture the full range of structural variation found across human populations. Comparing everyone to one mostly linear reference can make some variants easier to detect than others. The Human Pangenome Reference effort, therefore, began building a different kind of framework. Many high-quality genome sequences represented together. A first draft released in 2023 incorporated dozens of diverse individuals and added large amounts of sequence and structural variation missing from the traditional reference. The project has continued to expand, alongside other pangenome efforts around the world. A pangenome changes the metaphor. Instead of one road representing humanity, imagine a network of alternative paths through genomic regions. Some paths are shared by almost everyone. Others contain insertions, deletions, duplications, or rearrangements present in subsets of people. The reference itself becomes plural. Sequencing technology also became dramatically faster. The Human Genome Project cost billions of dollars as a multi-year international research program that built infrastructure and technology, as well as producing a reference. It is misleading to say that one ordinary genome simply cost $3 billion to read. But the contrast in scale is still remarkable. Today, high-throughput platforms can sequence a human genome for a tiny fraction of the original project's cost, and sequencing has become routine enough to be used in rare disease diagnosis, cancer care, pathogen surveillance, population studies, prenatal testing, and research across almost every branch of biology. Different sequencing technologies solve different problems. Short-read platforms can generate enormous numbers of accurate fragments efficiently. They are excellent for detecting many small variants, especially when aligned to a good reference. Long-read platforms can cross repetitive regions and reveal structural variants that short reads may miss. Nanopore sequencing takes a particularly direct approach. A DNA or RNA molecule passes through a tiny protein pore embedded in a membrane. Different combinations of bases alter the ionic current through the pore in characteristic ways. Software converts those changing current signals into sequence. A molecule becomes an electrical trace. The method can produce very long reads and can sometimes detect chemical modifications such as DNA methylation from the signal itself. Reading DNA, however, begins before sequencing. Researchers first need enough of the region they want to study, and one invention made that step extraordinarily easy. The polymerase chain reaction, PCR, was developed in the 1980s. PCR uses repeated cycles of temperature change to amplify a selected DNA region. First, heat separates the two DNA strands. Then the reaction cools enough for short synthetic primers to bind to sequences flanking the target. A heat-stable DNA polymerase extends from those primers, creating new copies. Repeat the cycle, and the number of target molecules can grow exponentially. 1 becomes 2, 2 become 4, 4 become 8. After many cycles, a region that was almost undetectable can be present in millions or billions of copies. PC are transformed molecular biology because it turns small amounts of DNA into analyzable amounts quickly. It became essential in cloning, diagnostics, sequencing, forensic analysis, evolutionary studies, and medical testing. Variations such as quantitative PCR allow researchers to monitor amplification and estimate starting amounts. Reverse transcription PCR first converts RNA into complementary DNA, allowing RNA viruses or gene expression patterns to be measured. The power of PCR also creates a problem. If the method can amplify a few molecules of target DNA, it can amplify contamination too. Ancient DNA laboratories therefore use strict cleanroom procedures, protective clothing, negative controls, and characteristic patterns of molecular damage to distinguish ancient fragments from modern contamination. Forensic genetics faces related challenges. A crime scene sample may contain DNA from several people, degraded fragments, or tiny quantities transferred indirectly. DNA evidence can be extraordinarily informative, but interpretation depends on sample quality, chain of custody, statistical models, and context. The discovery that transformed forensic genetics came from regions of the genome that vary greatly between people. Modern forensic profiling commonly examines short tandem repeats, or STRs. These are places where a short DNA motif repeats several times, with different people often carrying different repeat numbers. Looking across many independent STR loci produces a profile with strong power to distinguish unrelated individuals. A DNA match is not a magical statement that only one person in the universe could have produced a sample. It is a statistical comparison. Analysts estimate how probable the observed profile would be under competing hypotheses, using population frequency data and accounting for mixtures or relatedness when necessary. Identical twins can present a special challenge because their inherited STR profiles are usually the same. Whole genome or deep sequencing can sometimes detect post-zygotic differences, but ordinary forensic tests do not generally distinguish them. Genetic identification also raises ethical questions. A DNA sample reveals information not only about one person, but about biological relatives. Consumer genealogy databases have helped identify unknown remains and suspects in cold cases through distant relatives who voluntarily uploaded genetic data. The same capability raises questions about consent, privacy, law enforcement access, and how far one person's decision should expose family members who never submitted a sample. Genomics creates this kind of shared information repeatedly. If a clinical test finds a highly penetrant inherited variant, the result may have implications for siblings, parents, or children. A patient has a right to privacy, while relatives may have a strong interest in information relevant to their health. There is no enzyme that solves that conflict. Genetics became technologically powerful faster than society could make every ethical question simple. The Human Genome Project anticipated this problem by dedicating part of its funding to ethical, legal, and social implications. Sequencing the code was never going to be only a technical challenge. The code belonged to people. And as sequencing became cheaper, another surprise emerged. The most difficult part of genomics was no longer always reading the letters. It was understanding what they meant. A complete genome sequence contains millions of differences from a reference. Most are not disease-causing. That fact turns clinical genomics into an interpretation problem. Suppose a child has a rare disorder, and whole genome sequencing identifies several million variants. Which one matters? Researchers and clinical laboratories narrow the search using many kinds of evidence. Does the variant alter a protein coding sequence? Does it disrupt a splice site? Is it extremely rare in large population databases? Does the gene have a known role in the affected organ? Do other people with similar symptoms carry damaging variants in the same gene? Does the inheritance pattern fit the family? Has the variant been tested experimentally? Sometimes the answer is clear. A well-established pathogenic variant in a gene known to cause a matching disorder can end a diagnostic search that lasted years. Sometimes the answer is uncertain. A variant may be classified as a variant of uncertain significance because current evidence cannot show whether it contributes to disease. That label is not a polite way of saying harmless. It means the evidence is genuinely incomplete. As more genomes are sequenced, interpretations can change. A variant once thought suspicious may prove common among healthy people. Another may become convincingly linked to disease after more families are studied. Genomic medicine, therefore, depends on databases, data sharing, careful ancestry representation, and periodic reinterpretation. This is one reason diversity in genomic research matters. If reference databases contain disproportionately many people from one ancestry, variants common and harmless in underrepresented populations may be mistakenly treated as rare. Risk models developed in one population may perform worse in another because allele frequencies, linkage patterns, environments, and historical population structure differ. Better genomics requires better representation, not because people belong to sharply separate genetic groups, but because human variation is unevenly sampled across the world. The hardest variants to interpret are often outside protein coding regions. Only a small fraction of the human genome directly encodes protein sequences. Yet non-coding DNA contains promoters, enhancers, silencers, insulators, structural elements, origins of replication, non-coding RNA genes, and other sequences that influence how chromosomes work. A mutation in an enhancer can alter when a gene is expressed without changing the protein sequence at all. A structural variant can move an enhancer beside the wrong gene. A repeat expansion can disrupt transcription or create toxic RNA. A splice region variant can change which exons are joined. A mutation in a non-coding RNA can alter regulation far from its own genomic location. The old phrase junk DNA arose partly because so much non-coding sequence had no obvious function. Today we know that some non-coding regions are essential and others have measurable regulatory roles. But the opposite exaggeration is also misleading. Not every base in the genome has a specific selected function. Large portions are evolutionary remnants, repeated sequence, decayed mobile elements, or DNA whose effects may be weak, redundant, or unknown. Detecting biochemical activity at a sequence does not automatically prove that natural selection preserved that sequence for an important organism-level function. The genome contains function, noise, history, and material that may shift between those categories over evolutionary time. RNA expanded the picture even further. For decades, RNA was often introduced mainly as messenger RNA, transfer RNA, and ribosomal RNA. Those remain fundamental, but cells produce many additional RNA classes. MicroRNAs are short RNAs that guide regulatory complexes to messenger RNAs, often reducing translation or promoting degradation. Long non-coding RNAs can influence chromatin, transcription, nuclear organization, or RNA processing. Small interfering RNAs provide sequence-specific regulation in many organisms and inspired therapeutic technologies. RNA interference became a powerful example of biology turned into medicine. Researchers studying the nematode Cinorhabditis elegans found that double-stranded RNA could silence matching genes with remarkable specificity. The mechanism uses small RNA guides to direct protein complexes toward complementary messenger RNAs. Once understood, the pathway could be engineered. Several approved medicines now use small interfering RNA to reduce production of disease-related proteins, demonstrating again that genetic medicine does not always require changing DNA. Sometimes it is enough to change the flow of information temporarily. Antisense oligonucleotides use another strategy. These short synthetic nucleic acids bind selected RNA sequences. Depending on their design, they can trigger RNA degradation, block translation, or alter splicing. Some have been developed for rare neurological and muscular disorders. Spinal muscular atrophy provides a particularly elegant example. The disease is usually caused by loss of functional SMN1. Humans carry a related gene, SMN2, but a small sequence difference causes much of its RNA to be spliced in a way that omits an important exon. The antisense drug nusinersen changes that splicing pattern, encouraging SMN2 to produce more full-length functional protein. The treatment does not repair the original SMN1 mutation. It persuades a backup gene to be read differently. Genetics becomes medicine through an understanding of RNA processing. Pharmacogenomics asks another practical question. Can inherited variants help predict how a person will respond to a drug? Some variants alter drug metabolizing enzymes. Others change transporters, receptors, immune recognition, or drug targets. In selected cases, genetic testing can help guide drug choice or dose or prevent severe adverse reactions. The HLA system provides dramatic examples. Certain HLA alleles can greatly increase the risk of dangerous immune reactions to specific medicines. Testing before treatment can prevent those reactions in populations where the relevant alleles are common enough to justify screening. But pharmacogenomics is not a universal decoder for the perfect medication. Many drug responses depend on kidney and liver function, age, interactions with other drugs, diet, adherence, disease state, and variants across multiple genes. Genetics can contribute one layer of information without replacing clinical judgment. The same principle applies to prenatal and reproductive genetics. Chromosome screening, carrier testing, prenatal diagnosis, and pre-implantation genetic testing can identify selected genetic conditions before birth. These technologies can reduce uncertainty or help families avoid severe inherited disease. They also create decisions that cannot be settled by molecular biology alone. A test can estimate whether a fetus carries a trisomy. It cannot tell a family what values should guide a pregnancy decision. A carrier screen can identify reproductive risk. It cannot decide whether a person should become a parent. An embryo test may detect a known familial mutation. Extending embryo selection toward complex traits introduces much weaker prediction and much larger ethical questions. Genes can provide information. People still have to decide what that information means in a life. The distinction becomes especially important when talking about genetic risk. A pathogenic variant with very high penetrance can make disease likely, but penetrance may still vary with age, sex, modifiers, and environment. A common risk allele may raise probability only slightly. A polygenic score may shift a statistical distribution without predicting one person's future with certainty. The phrase "you have the gene for a disease" is therefore often too crude. Everyone has genes involved in cancer, diabetes, heart disease, and brain function. What differs are variants, copy numbers, regulatory states, and combinations. The genome is not divided into normal genes and disease genes. The same gene can support normal biology in one state and contribute to disease when altered. That idea becomes crucial when we move from reading genomes to editing them. If a disease is caused by a DNA sequence, could we change that sequence? For most of genetic history, this was a thought experiment. Then bacteria revealed that they had already evolved molecular tools capable of recognizing and cutting DNA with extraordinary specificity. CRISPR began not in a gene therapy clinic, but in the genomes of bacteria and archaea. Researchers studying microbial DNA noticed unusual repeated sequences separated by variable spacers. The repeats became known as clustered regularly interspaced short palindromic repeats, or CRISPRs. The spacers turned out to contain fragments related to viruses and other mobile genetic elements. A bacterium surviving infection could acquire a piece of an invader's DNA and place that sequence into a CRISPR array. The array could later be transcribed into guide RNAs. Together with CRISPR-associated proteins, those RNAs helped recognize and destroy matching genetic material during another encounter. It was an adaptive immune system built from nucleic acid memory. Different microbes evolved many versions of CRISPR defense. One system, from Streptococcus pyogenes, used a protein called Cas9 guided by RNA to cut DNA. Emmanuel Charpentier's work on a small RNA called tracrRNA helped reveal how that system functioned. Jennifer Doudna, a biochemist with deep expertise in RNA, joined Charpentier in asking whether the components could be simplified and reprogrammed. In 2012, their collaboration showed that Cas9 could be directed by designed RNA to cut a chosen DNA sequence in vitro. Soon, several groups demonstrated CRISPR editing in eukaryotic cells. The transformation was rapid because the system was relatively easy to retarget. Change the guide RNA sequence, and the same Cas protein could be aimed at a new genomic location, provided the target sat beside a short recognition sequence called APAM. Cas9 does not rewrite DNA by itself. It makes a break. The cell's own repair machinery creates the edit. If the goal is to disrupt a gene, researchers can take advantage of end joining, which often introduces small insertions or deletions as the break is repaired. If a precise sequence change is desired, a repair template can sometimes guide homology-directed repair, although efficiency depends strongly on cell type and state. CRISPR rapidly became a research tool. Scientists could disable genes one by one to discover their functions. They could build models of disease. They could engineer immune cells, crops, microorganisms, and laboratory animals. Large CRISPR screens could test thousands of genes in parallel to find which ones a cancer cell depends on or which ones influence viral infection. Then gene editing entered medicine. The first FDA-approved treatment using CRISPR-based genome editing was Casgevy, approved in December 2023 for certain patients with sickle cell disease and subsequently approved for transfusion-dependent beta thalassemia. The treatment is performed outside the body. Doctors collect blood-forming stem cells from the patient. In the laboratory, CRISPR-Cas9 edits a regulatory region controlling BCL11A in those cells. BCL11A normally helps suppress fetal hemoglobin after birth. Reducing its activity in the relevant blood cell lineage allows more fetal hemoglobin to be produced. This is an elegant detour around the original disease mutation. The therapy does not simply replace the sickle beta globin base with the typical sequence. Instead, it reactivates a developmental hemoglobin program that can compensate for defective adult hemoglobin. The edited stem cells are returned to the patient after conditioning treatment makes space in the bone marrow. The process can be transformative, but it is not a simple injection of molecular scissors. Collecting stem cells, editing them, preparing the patient with chemotherapy, returning the cells, and managing infection and other risks requires specialized medical care. Long-term follow-up is important. Gene editing can reduce one source of disease while still carrying procedural and biological risks. That balance has always shaped gene therapy. The first modern human gene therapy trials began around 1990 with enormous optimism. Researchers hoped that if a disease came from a faulty gene, adding a functional copy might correct the problem. Some early results were encouraging. Then serious setbacks changed the field. In 1999, 18-year-old Jesse Gelsinger died after receiving an experimental adenoviral gene therapy in a clinical trial for ornithine transcarbamylase deficiency. He developed a severe systemic inflammatory response and organ failure. The death led to intense scrutiny of trial design, disclosure, vector biology, and oversight. Soon after, several children treated for X-linked severe combined immunodeficiency developed leukemia after therapeutic retroviral vectors integrated near genes controlling cell growth. The treatment had restored immune function, but insertional activation of oncogenes created a new danger. Gene therapy did not disappear. It became more cautious. Researchers redesigned viral vectors, improved control of gene expression, studied integration patterns, developed self-inactivating systems, and expanded long-term monitoring. Years later, gene therapy returned with treatments capable of producing dramatic benefits. Luxturna delivers a functional RPE65 gene to retinal cells in people with certain inherited retinal dystrophies. Zolgensma delivers a functional SMN1 gene for spinal muscular atrophy. Other therapies modify a patient's own blood stem cells or immune cells outside the body and return them. The word gene therapy now covers several strategies. Gene addition supplies a functional gene without necessarily repairing the original sequence. Gene editing changes DNA at a selected locus. RNA therapies alter messages without permanently changing the genome. Engineered cells can receive new receptors or regulatory circuits. Each approach carries different benefits and risks. CRISPR itself has evolved beyond the original Cas9 double-strand break. Base editors combine a DNA targeting system with enzymes that chemically convert one base into another within a small editing window. They can make selected substitutions without creating a full double-strand break. Prime editing uses a modified Cas enzyme and reverse transcriptase together with a specialized guide, RNA, that encodes the desired change. In principle, it can install a broader range of substitutions, insertions, and deletions. These technologies are powerful research and therapeutic platforms, but precision does not mean perfection. Editors can affect unintended sites. Even at the intended site, repair outcomes can vary. Delivery to the correct tissue remains difficult. An edit that works efficiently in blood stem cells may be far harder to deliver safely throughout brain, muscle, or lung. The largest challenge in genetic medicine is often not knowing what sequence to change. It is reaching enough of the right cells without harming the rest. Blood is unusually accessible. Stem cells can be collected, edited, tested, and returned. The liver is also relatively accessible to certain delivery systems because it naturally takes up particles from blood. The brain is protected by anatomical barriers. Skeletal muscle occupies an enormous mass. The eye is small and compartmentalized, which can make local delivery attractive. Anatomy becomes part of genetics. There is also a boundary between editing a patient and editing future generations. Somatic gene therapy changes cells in one treated person and is not intended to be inherited by children. Germline editing would alter eggs, sperm, or embryos in ways that could pass the change to future generations. In 2018, Chinese researcher He Jiankui announced the birth of children whose embryos had been edited with CRISPR in an attempt to alter CCR5, a gene involved in HIV entry into cells. The experiment was widely condemned. The medical need did not justify the risks. The edits were not guaranteed to reproduce a naturally protective variant. Mosaicism and unintended changes were concerns. The children could not consent. And because the changes were heritable, consequences could extend beyond the individuals directly involved. The episode made a central ethical distinction impossible to ignore. The ability to alter DNA does not answer whether a particular alteration should be made. Therapeutic editing for a severe disease with no good alternatives is one question. Editing embryos to select or enhance traits is another. And complex traits are especially poor candidates for the fantasy of precision design. Height, intelligence, athletic performance, personality, disease susceptibility, and most other human characteristics arise from large networks of genes and environments. Variants with beneficial associations in one context can have unwanted effects in another. Genetic correlations mean that changing a pathway for one trait can influence several others. There is no simple menu of human features encoded by independent switches. The genome is interconnected. That is why the most successful genetic medicines often target mechanisms we understand unusually well. A missing enzyme, a toxic protein, a blood cell receptor, a defined splice defect, a regulatory element controlling hemoglobin. Precision grows from knowledge, and knowledge grows slowly, one mechanism at a time. Even now, after millions of human genomes have been analyzed in research and clinical settings, many genes remain incompletely understood. Many variants cannot be interpreted confidently. Many non-coding regions have effects we can detect statistically, but cannot yet explain mechanistically. The genome was sequenced before it was understood. In many ways, that is still true. There is one more reason not to imagine the genome as a sacred text that every cell preserves word for word. Some cells deliberately rewrite parts of it. Your adaptive immune system depends on this controlled genetic instability. B cells and T cells must recognize an enormous variety of possible pathogens, far more than could be encoded by one inherited gene for every antigen receptor. During their development, these lymphocytes assemble receptor genes from separate DNA segments through a process called VDJ recombination. Proteins called RAG1 and RAG2 cut DNA at selected recombination signals. Different variable, diversity, and joining segments are brought together. Bases can be removed or added at the junctions. Separate receptor chains are paired. The result is a vast repertoire of receptor sequences created somatically rather than inherited in finished form. A mature T cell can therefore contain a rearranged receptor gene that did not exist in that exact arrangement in the fertilized egg. B cells go even further after activation. Inside germinal centers, an enzyme called activation-induced cytidine deaminase introduces mutations into antibody variable regions. B cells with improved binding can be selected and expanded. The immune system intentionally concentrates mutation in one genomic region to refine molecular recognition. It is a controlled risk. The same DNA cutting and mutating machinery can contribute to lymphoma if it acts at the wrong site, or chromosome rearrangements join growth, promoting genes to active regulatory regions. A system useful enough to defend the body is dangerous enough to require tight control. Genetics repeatedly returns to this pattern. Stability and change are both necessary. Meiosis illustrates the same balance from another direction. Chromosomes usually segregate with extraordinary reliability, but not always. If homologous chromosomes fail to separate in meiosis 1, or sister chromatids fail in meiosis 2, a gamete can receive an extra chromosome or lack one. This is non-disjunction. After fertilization, such errors can produce aneuploidy, a chromosome number different from the usual set. Most severe autosomal aneuploidies are not compatible with development to birth. Some are. Trisomy 21, the cause of Down syndrome, is the best known example. Instead of 2 copies of chromosome 21, most cells contain 3. The effect is not caused by one abnormal gene. It is a dosage change affecting many genes at once. The probability of certain meiotic chromosome segregation errors rises with maternal age, partly because human oocytes begin meiosis before birth and can remain arrested for decades before completing it. Proteins and chromosome structures responsible for maintaining alignment and cohesion must function across an unusually long biological interval. Again, time becomes part of genetics. Mitochondrial genetics has its own unusual rules. Each cell can contain many mitochondria, and each mitochondrion can contain multiple copies of mitochondrial DNA. A person may therefore carry a mixture of different mitochondrial genome variants within the same cell or tissue, a state called heteroplasmy. When cells divide, mitochondrial genomes are partitioned among descendants. The proportion of a disease-associated variant can drift upward or downward in different tissues. Symptoms of mitochondrial disease can depend on whether the fraction of altered mitochondrial DNA crosses a functional threshold in organs with high energy demands. Two siblings inheriting the same mitochondrial mutation from their mother can therefore be affected differently. The inheritance is maternal, but the outcome can be probabilistic. Mitochondria also remind us that the genome inside the nucleus is not the only genetic system in the cell. Most genes needed for mitochondrial function are actually nuclear genes. Proteins encoded in the nucleus are synthesized in the cytoplasm and imported into mitochondria. A small mitochondrial genome works inside a much larger nuclear partnership whose roots reach back to an ancient endosymbiosis between early eukaryotic cells and bacteria. Even the existence of two genomes inside one human cell is an evolutionary fossil. Modern technology is beginning to reveal variation at the level of individual cells rather than averaging an entire tissue. For most of molecular biology's history, a sample meant a mixture. Extract RNA from a piece of liver and the result blends hepatocytes, immune cells, blood vessel cells, fibroblasts, and other populations. Sequence tumor DNA and the signal mixes different cancer clones with normal cells. Single-cell technologies change that perspective. Single-cell RNA sequencing measures gene expression profiles from thousands or millions of individual cells. Single-cell DNA methods can identify mutations and copy number changes in separate cells. Chromatin accessibility assays reveal which regulatory regions are open. Spatial methods add another dimension by preserving where cells were located inside tissue. A tissue stops looking like one average cell. It becomes a community of states. This has transformed developmental biology and cancer research. A tumor that seemed uniform under a microscope may contain proliferating clones, exhausted immune cells, stromal populations, hypoxic regions, and rare resistant cells. A developing embryo contains transient states that exist only briefly before becoming another cell type. Genetics is moving from sequence toward context. Which cell carried the variant? Which genes were active at that moment? Which neighbors were present? What chromatin state surrounded the gene? What happened before and after? The questions are becoming more dynamic even as the DNA alphabet remains the same for letters. Artificial intelligence and large computational models are also entering genomics because the data have become too large for manual interpretation. Algorithms can predict how a DNA variant may alter protein structure, splicing, transcription factor binding, or gene regulation. Models trained on vast collections of biological sequences can identify patterns no person could inspect directly. These systems can narrow experiments and generate hypotheses. They do not remove the need for experiments. A computational score is evidence, not biological truth. A model can learn biases in its training data. It can be confident about regions for which biology is poorly represented. Predictions still need validation in cells, organisms, populations, and ultimately patients when medical decisions are involved. The history of genetics is full of moments when measurement became easier than interpretation. Mendel could measure phenotypes before genes were visible. Chromosome maps existed before DNA sequence could be read. The human genome was sequenced before most variants could be understood. Single-cell atlases can now contain millions of cells before we know what every state means. More data does not automatically create more explanation, but it gives science a finer place to ask questions. And perhaps the most important question remains the oldest one. What does the genome actually mean for a person? A genome can tell you many things. It can reveal a variant that changes an enzyme. It can show that a chromosome segment came from a particular ancestral population. It can identify biological relatives. It can estimate susceptibility to selected diseases. It can reveal that a tumor has lost a repair pathway or that a child carries a mutation explaining years of unexplained symptoms. What it cannot do is contain a complete person in 3 billion letters. There is no nucleotide sequence for one specific memory of a summer evening. No isolated base that determines whether someone becomes patient, generous, fearful, curious, musical, lonely, or brave. Genes influence the development of brains and bodies in which those qualities emerge, sometimes strongly. But the path from sequence to person travels through development, family, culture, nutrition, illness, learning, accident, relationships, opportunity, stress, sleep, hormones, chance, and time. Even the brain is not built by DNA alone in the way a machine is assembled from a fixed diagram. Genes establish developmental rules. Growing neurons respond to chemical gradients and neighboring cells. Synapses form in enormous numbers. Activity strengthens some connections and weakens others. Hormones change developmental trajectories. Experience changes circuits. Injury changes them again. The genome helps build a system capable of being changed by the world. That capacity for change is itself biological. The false choice between nature and nurture dissolves here. Nature built an organism that can be nurtured. Genes encode proteins that detect environments, respond to environments, learn from environments, and sometimes alter environments. Culture changes diet and disease exposure, which changes selection. Agriculture changed human genomes by favoring variants related to starch digestion, milk consumption, immunity, and metabolism. Pathogens shaped immune genes. Human behavior became part of the environment acting back on human evolution. Genes and culture have been interacting for thousands of years. The genome is therefore neither a rigid destiny nor an empty page. It is a set of inherited constraints and possibilities carried into a life that continues to unfold. This is also why two people can share a disease-associated variant and have different outcomes. Penetrance may be incomplete. Other variants modify risk. A protective exposure may matter. A harmful exposure may matter. Random developmental events can matter. Medical treatment changes trajectories that evolution never anticipated. And this is why a person can carry a mutation without being defined by it. Medical language has slowly learned this lesson. A patient is not a defective genome. A disability is not a sequence error in the moral sense. A variant can cause disease while the person carrying it remains much larger than the mechanism being studied. Genetics can describe biology with extraordinary precision. It cannot assign human worth. History gives good reasons to say this clearly. Long before DNA was understood, ideas about heredity were misused to rank populations and justify coercive eugenics programs. In the late 19th and 20th centuries, governments and institutions in several countries used claims about inherited fitness to support forced sterilization, marriage restrictions, segregation, immigration policies, institutionalization, and, in Nazi Germany, mass murder. These policies were scientifically crude and ethically catastrophic. Modern genomics has shown human variation in exquisite detail, but greater technical precision does not automatically protect against old mistakes. Genetic differences can be real without forming a hierarchy of human value. Statistical differences between populations do not define individuals. A heritable trait is not a moral trait. A risk estimate is not a verdict. Good genetics requires humility about what DNA can and cannot explain. It also requires privacy. Unlike a password, a genome cannot be changed after a data breach. It can reveal information about relatives. Future science may extract meanings from stored sequences that were unknown when a sample was first collected. Consent for genomic data therefore reaches forward into uncertainty. Researchers have developed systems for data identification, controlled access, consent, and governance, but no system eliminates every risk. The value of genomic research depends on people being willing to contribute data, and that trust depends on responsible use. The future of genetics will therefore be shaped not only by faster sequencers or more precise editors. It will be shaped by decisions about fairness, access, consent, disability, ancestry, privacy, and who benefits from the technologies being built. A therapy that can cure a genetic disease but is available only to a tiny fraction of affected people solves one molecular problem while leaving a social one. A risk model trained on one ancestry can deepen inequity if applied carelessly to everyone else. A database can accelerate discovery and still require strong protection for the people represented inside it. The science becomes more powerful, and the responsibility grows with it. For all of that complexity, the molecule itself remains surprisingly simple. For bases, A pairs with T, G pairs with C. Two strands run beside one another. From those simple rules come bacterial chromosomes, oak trees, whales, fungi, hummingbirds, and human brains. Not because the same four letters contain some hidden human quality, but because sequence length, regulation, duplication, mutation, selection, development, and immense evolutionary time can build complexity from a small alphabet. Your genome contains approximately 3 billion base pairs in one haploid set, but the number alone is not especially impressive in biology. Some organisms have genomes much larger than ours. A larger genome does not imply a more intelligent or more complex organism. Much of genome size reflects repeats, duplications, transposable elements, and lineage-specific history. Humans do not have the most genes. We do not have the most DNA. Our biology depends on how genetic systems are regulated and integrated, not on winning a contest of molecular quantity. Even the concept of a gene becomes less sharp the closer we look. Where does a gene begin if several promoters can be used? Where does it end if alternative polyadenylation creates different transcripts? Is a regulatory enhancer part of the gene it controls if it lies hundreds of thousands of bases away? What about an RNA gene that never makes protein? What about overlapping transcripts made from opposite DNA strands? The word gene remains useful. It is simply no longer a tiny bead on a chromosome carrying one trait. Modern genetics replaced that old image with networks. And networks are harder to summarize. Perhaps that is why the double helix remains such a powerful symbol. It gives us one clean shape inside a science that became increasingly complicated after the shape was discovered. The helix is real. The meaning around it is layered. Now, near the end of our journey, we can return to the cell where we began. Hours may have passed in the outside world, but inside the cell the work remains microscopic. Replication origins have fired across its chromosomes. Forks have moved in both directions. Polymerases have added complementary bases. Okazaki fragments have been joined. Proofreading enzymes have removed mismatches. Repair pathways have corrected damage. The copied chromosomes are not naked strings. New histones have been assembled with old ones. Chromatin marks are being restored. Regions that were active before replication need to recover their regulatory state. Loops and domains will be rebuilt. The daughter cells must inherit not only DNA sequence, but enough molecular organization to continue being the kind of cells they were. A cell division is therefore both copying and remembering. The chromosomes condense. Microtubules attach to structures at their centromeres. Sister chromatids align. Checkpoint proteins ask, chemically, whether attachment is correct. Then the sisters separate. One set moves toward one side of the cell. One set moves toward the other. New nuclei form. The cell divides. Two cells now occupy the space where one cell had been. Their genomes are almost the same. Almost. Perhaps one base changed during the process and escaped repair. Perhaps nothing functionally important changed at all. Perhaps a methylation pattern differed slightly. Perhaps one daughter received more of a particular protein or organelle than the other. Life continues without perfect copying because it has never required perfect copying. It requires copying accurate enough to preserve function and flexible enough to allow change. Elsewhere in your body, another cell is not dividing. A neuron may remain in a largely non-dividing state, while transcribing genes needed to maintain membranes, receptors, transport systems, and synapses. A muscle fiber is producing proteins that repair ordinary wear. A liver cell is changing gene expression in response to nutrients and hormones. Bone marrow is producing new blood cells. Immune cells are surveying tissues with receptors assembled through controlled DNA rearrangement. Some cells are turning genes on. Some are turning them off. Some are destroying RNA molecules that are no longer needed. Some are replacing proteins that lasted minutes. Others are maintaining proteins and structures that can persist much longer. DNA does not perform all this work alone, but it remains quietly available at the center of most nucleated cells, a chemically stable reference that the rest of the system can return to again and again. The sequence you inherited has already survived an extraordinary journey. Before it was yours, portions of it were copied through your parents' cells. Before that, through their parents. Recombination rearranged the pieces every generation. Mutations appeared. Some disappeared. Segments moved between ancient populations. Viral sequences entered chromosomes. Duplications created extra copies. Selection preserved some variants and removed others. Drift changed frequencies by chance. Trace any one base far enough backward and the idea of your DNA becomes strange. It was never waiting for you. It passed through organisms that could not know you would exist. Some sequence near a gene may have been present before our species appeared. Some coding regions are recognizably related to genes in other mammals. Some cellular machinery reaches back to the earliest eukaryotes. The logic of DNA replication and the genetic code reaches deeper still, toward common ancestors shared by all known cellular life. The code inside you is ancient not because your personal sequence is unchanged, but because the system of inheritance is older than animals, older than plants on land, older than complex multicellular bodies. A modern human genome is one temporary arrangement in a 4 billion-year experiment in copying with variation. And for all the attention we give genes, much of what the genome does successfully is invisible. A repair pathway fixes a lesion before it becomes a mutation. A cell with dangerous damage stops dividing. A developmental gene activates at the right place and then becomes silent. A chromosome segregates correctly. A protein folds well enough. A regulatory circuit keeps an enzyme within the range the cell needs. The absence of genetic disease is not the absence of genetics. It is genetics working so routinely that nothing draws attention to it. Perhaps that is the quietest wonder of DNA. Not that it makes us special. Not that it contains a secret explanation for every part of a person. But that trillions of living cells can preserve, read, repair, and reinterpret in ancient molecular language every day without a central reader overseeing the process. There is no tiny librarian inside the nucleus. No molecule understands the genome. Polymerases respond to chemical fit. Transcription factors bind particular shapes and sequences. Ribosomes pair codons with transfer RNAs. Repair enzymes recognize distortions. Chromosomes move through forces generated by proteins. Meaning emerges from interactions. The cell works because the pieces affect one another in reliable ways. And so the code inside you is not like software running on biological hardware. The code, the reader, the machinery, and the environment are all made of the same living chemistry. DNA is read by proteins whose sequences were influenced by DNA. Those proteins regulate the DNA that helps produce more proteins. RNA sits between them, but also regulates both. Membranes create compartments. Metabolism supplies energy. Signals from outside the cell change what the genome does next. The system closes around itself. A living cell is not a book being read. It is a process continuing. Tonight, while you rest, that process does not stop. Some cells will divide. Others will repair damage. Some genes will become more active according to circadian rhythms and hormones. Others will become quieter. RNA molecules will be made and destroyed. Proteins will be assembled. DNA will remain folded inside chromatin, protected but accessible, waiting wherever the cell needs it. You do not need to remember the names of the enzymes. You do not need to picture every chromosome. You do not need to keep track of adenine or cytosine, of promoters or enhancers, of telomeres or repair pathways. The cells do not know those names either. The names belong to us, because naming is how human minds make a vast, invisible process easier to follow. The process was here before the vocabulary. It will continue after attention drifts away from it. Two strands paired by simple chemistry. Billions of bases folded into microscopic space. Copies inherited from parents and altered by generations. Ancient viral fragments beside human genes. Messages made from selected regions and proteins built from those messages. Mutations appearing quietly, most of them passing unnoticed. Repair occurring even more quietly. A genome that can influence a life without containing the whole life inside it. The cell where we began has finished dividing now. Its daughter cells return to ordinary work. The chromosomes loosen from their most compact form. Nuclear pores exchange molecules with the cytoplasm. RNA polymerases find active genes. Ribosomes continue translating messages. DNA repair proteins continue their patrol. Nothing about the scene looks like a code from the cell's point of view. There are only molecules interacting with molecules. And yet from those interactions come inheritance, development, memory of evolutionary history, and the continuity of life from one generation to the next. The double helix can remain folded in the dark. The chromosomes can remain inside their nuclei. The old sequences can continue their patient copying. You do not have to supervise any of it. The code inside you has been traveling for billions of years. For tonight, it can continue without your attention. Sleep well. Good night.